Now let's talk about the another question on the screen, that is question number 14. What is point mutation? Also give one example. So first of all, we have to understand what is mutation. So mutation is interchange, deletion or insertion. Interchange, insertion or deletion in a nucleotide basis of a DNA is known as mutation. When we're talking about point mutation, then this point mutation is a mutation when mutation is happening in only single nucleotide base. When mutation is taking place only single nucleotide base of a segment of DNA, then this type of mutation is known as point mutation. Then this mutation is known as point mutation. This point mutation can be done by addition of a single nucleotide base. It can also occur with deletion, with addition, with interchange of a single nucleotide. Single nucleotide when is inserted, deleted or interchanged in a segment of DNA, that mutation is known as point mutation. Alright, and if we talk about example, then for example we have sickle cell anemia. Sickle cell anemia, our erythrocytes, our red blood cells are having a gene, a normal gene which is known as HBB gene. HBB gene have so many nucleotide bases. On the 17th nucleotide base, we have GAG. We have a codon named as GAG which codes for amino acid known as glutamic acid. It is interchanged. The GAG, the codon GAG, the A between G and G, the A is interchanged by T. The A is interchanged by T. And this is causing a codon, this is changing a codon into GTG, which is coding now, which is making amino acid as valine. And this amino acid valine is producing sickle cell anemia in an individual. So now let's write answer of question number 14. What is point mutation? Before going on to point mutation, we will write a simple basic definition for mutation. So, interchange, deletion and addition in the nucleotide basis of a segment of DNA, then this phenomena is known as mutation. This phenomena is known as mutation. When we are talking about point mutation, these things, interchange, deletion or addition, is going to happen only in single nucleotide base of a segment of DNA. So the mutation, we know now that what is mutation. So now mutation of single nucleotide, mutation of a single nucleotide base of a segment of DNA is known as point mutation. Is known as point mutation. All right. So if we're talking about example, we have sickle cell anemia as an example. Sickle cell anemia. What happens in sickle cell anemia? Suppose we have HBB gene, which is a normal gene for our erythrocytes, for our red blood cells. We have a normal gene named as HBB gene. HBB gene have so many nucleotide base pairs. So on the 17th nucleotide, we have a codon On the 17th nucleotide, we have a codon GAG. GAG. In a normal condition, we have a codon named as GAG, which is coding or producing amino acid known as glutamic acid. Glutamic acid, which is a normal. And when point mutation occurs in any individual, what happens? This simple normal codon having GAG, this A is replaced by T and making it this codon GTG. A is replaced by T, making the codon GTG. Now point mutation occurred. The interchange of one nucleotide base has happened. The interchange of one nucleotide base has happened in a segment of DNA. A is replaced by T. So this codon is now going to produce another type of amino acid which is known as valine, which is an abnormal condition. And this condition, when reaches into an individual, this produces the erythrocytes in a sickle shape, and this disorder is known as sickle cell anemia. So this is all about question number 14, and this is the complete solution of it.